take that as a yes. So like Dr. Knapp said, be presenting today on the effect of sudden death syndrome on seedling emergence. To give you a little brief outline today about what I'm going to be talking about, kind of the background about sudden death syndrome, just a little bit about the disease and how it's caused, a little bit about the causal organism, the disease cycle and the epidemiology of the disease, the symptoms that foliar, above ground and foliar symptoms that the disease is known to produce, and then current management practices that are in place that are affecting sudden death syndrome disease development, and then some of the factors that are influencing the infection of the causal organism, and then some about my research, and then I'll have questions at the end. So here's just a picture, kind of a general picture or a basic idea of what SDS looks like on soybeans. I'm sure most of us have seen it being in Iowa, driving around in late August. We typically see this disease occur. And I'll explain a little bit more about this, about these symptoms anyway, in a minute. So the movement of sudden death syndrome first became really relevant in 1971 when it was first observed in Arkansas. That was discovered by Roop and Roy et al. And then from then on, it's progressively moved northward and north and to the east and into Kentucky, Illinois, Indiana, all the major soybean production areas of the Midwest. And then finally, it was first confirmed case was in Iowa. It was in 1993 by X.B. Yang. And then 1998 was finally what they termed, or a pathologist termed, as an epidemic. And that occurred when over a third of the counties in Iowa were actually had developed cases of, confirmed cases of SDS. And then today, kind of jumping forward to what today's system, or current day, today there's a problem actually in really the southern 2/3 of the state. If you can kind of draw a line from where Highway 20 splits the state, to the north would be where we typically see a lot of white mold infections, and below the Highway 20 line is where we typically see the development of SDS infections. So SDS is caused by the causal organism Fusarium solani, and this was termed by Roy in 1989 and then Roop in 1989 as well. These are Roy and Roop are two major pathologists that work in the University of Arkansas. And with how they determine the nomenclature is once they determine what the disease is through a process called Koch's postulates. And what Koch's postulates is when the disease is first observed, they isolate the disease, and then we'll take that back, grow it on culture of a medium of some sort, say PDA, and then they'll re-inoculate that into a plant, in this case soybeans, and see if the same disease symptoms are developed later on throughout the growth stages of the plant. And if the same disease symptoms are found, then they know they have isolated just the single organism that's causing that disease. So that's how Fusarium solani was finally found to be the causal organism of SDS. So then as time progressed and technology was able to advance, things like that, in 1997, Roy et al. Discovered that it was further the nomenclature of the causal organism to be Fusarium solani formae specialis glycines. Basically what the specialis glycines means is that sudden death syndrome only affects soybeans. Soybeans being glycine max kind of comes along with it, that sudden death syndrome can only affect soybeans. So then in 2003, Aoki et al., through the use of quantitative PCR and DNA technology, actually found that it finally determined that the final name was going to be Fusarium virguliforme, which it currently stands as now. So in order to understand sudden death syndrome, you need to understand the disease life cycle of it. So obviously when you have a soil-borne pathogen, it obviously either resides in crop residue or the soil surface. And then with SDS, it actually resides in survives in survival structures called the chlamydiospores. With these chlamydiospores, and then in the spring, once conditions or environmental conditions are right, so soil moisture and soil temperature conditions are conducive for such, it'll actually infect the hyphae of the chlamydiospores, will then infect the soybean root And this is kind of where discussion or a lot of research is being done right now, is they don't really know or understand when the actual infection is occurring. There's unpublished data out right now that shows that the disease can actually, or the causal organism can actually infect as early as 1 day after germination, or it can affect 3 weeks to 10 days, even as far along as 6 weeks into emergence. So there's a lot of discrepancy as to when the disease actually becomes, or when the causal organism infects the soybean root. So from then on, once it's actually infected into the soybean root, the hyphae then colonize the soybean root, therefore moving up and down throughout the root system, the hyphae do. And then eventually then we get disease symptom development, and we get SDS. So there are also two important things to remember with SDS. There's throughout the disease cycle, in order to get infection of Fusarium virguliforme at planting, this occurs obviously at planting and early vegetative growth, but then you also need high soil moisture and cool soil temperatures. And this was discovered by Roy et al. In 1997, but also work has been done here in Iowa State by X.B. Yang and his laboratory across the street in plant pathology. And then once infection has occurred, then you need to have disease symptoms occur. And typically the disease symptoms occur during the seed filling process, so the reproductive stages of the soybean plant. And this is typically seen, the highest disease symptoms are often seen with warmer temperatures and high soil moisture. So if you can kind of think around, say, state fair weather, as we would call it here in Iowa, you get hot, humid, muggy days, and then you also get a pop-up rain shower. That's when we typically see the highest amounts of SDS starting to develop, disease symptoms to develop. So now we'll kind of move on to a little bit about the disease symptoms that it causes. So once infection has occurred in the root, more as the foliar severity increases of the disease, root symptoms also increase. Root disease symptoms occur. So then the typical symptoms that we see are often discoloration of the root. This can be either external or internally. And then we also have a blue sporulation, and I don't know if you can kind of see it there in the picture, but there's a blue mass that the Fusarium virguliforme produces on the soybean root. That's also often a good indication that you know you have sudden death syndrome developing. And then as the disease progresses and it progressively becomes worse, you get root necrosis and reduced root volume. Again, this is also going to start to affect soybean yield because you're not being able to take up as much water and essential nutrients that are then you're going to equate to soybean yield. So once the fungus has actually infected the soybean root, pathologists had a really hard time being able to isolate the disease. And this comes from the discovery finally by Jen et al. In 1996 that actually a phytotoxin was being produced from the ground up, or from basically the soil surface up. This phytotoxin was being produced and then would actually cause the foliar symptoms of the disease. Once this phytotoxin was identified, and then a polypeptide was identified, and then right now research is going on downstairs, and Dr. Madon, I murder his last name always. He's working with identifying more about the polypeptide and how to possibly make it so that foliar symptoms don't develop because of it. And then foliar symptoms are independent of root symptoms. So what we mean by this is that you could actually inoculate, sometimes you can inoculate the below ground portions of the root and not ever observe foliar symptoms. But if or vice versa, you can inoculate the top or the foliar portion of the plant and never observe root symptoms. It's one of those things that's very hard to study about this disease. So moving from obviously the ground up to the root, or from the roots up to the shoots, now that the phytotoxin has moved up through the xylem system, then we get foliar symptoms. And typical foliar symptoms occur early on in the growing season, around typically R1, so the first reproductive stages of the soybean plant. And these symptoms are just really chlorotic spots, as you can see there in the top right, or top left, excuse me. And then you also get leaf mottling. And then as the disease progressively becomes worse and worse, then we start to see interveinal chlorosis and necrosis.
Thank you for watching the Agronomist Podcast. YouTube limits video lengths to 10 minutes. You can watch this seminar in its entirety at the Department of Agronomy website, www.agron.iastate.edu.